Have you ever wondered how real-time PCR works? Well, let's delve into it. Real-time PCR, also known as quantitative polymerase chain reaction or qPCR, is a technique that monitors the progress of a PCR reaction as it occurs in real time. It quantifies a relatively small amount of DNA, cDNA, or RNA product. The process is based on the detection of fluorescence produced by a reporter molecule, which intensifies as the reaction proceeds. This technique is a powerful tool in molecular biology, allowing for the exponential amplification of DNA sequences. Unlike conventional PCR that requires post-reaction analysis using gel electrophoresis, real-time PCR saves time by providing analysis during the process. This is where the term real-time stems from, signifying its ability to monitor the amplification process as it happens rather than after completion. Now that we know what real-time PCR is, let's understand how it works. Understanding the process of real-time PCR is crucial to appreciate its significance. Let's dive into the step-by-step process of real-time PCR. The first thing we need is a pair of primers. These primers are nothing but short DNA sequences that are complementary to the DNA sequence we are interested in amplifying. Now, these primers attach themselves to the DNA strand, acting as the starting point for DNA synthesis. The DNA polymerase, an enzyme that synthesizes new DNA, comes into play next. It starts extending the primers, adding nucleotides one by one to form a new DNA strand. This results in the formation of a PCR product, which could be DNA, cDNA, or RNA, depending on the starting material. These newly formed strands, also known as amplicons, act as templates for the next cycle. The same primers are used, and the DNA polymerase extends them again, leading to an exponential amplification of the DNA molecules. This cycle repeats itself over and over, doubling the number of DNA copies with each cycle. The beauty of real-time PCR lies in its ability to monitor this amplification process in real time. Unlike conventional PCR, where you have to wait until the end of the process to analyze the results, real-time PCR provides a continuous reading of the amount of DNA present in the sample. This is achieved by the use of a reporter molecule that emits fluorescence. As the PCR reaction proceeds, the amount of reporter molecule increases, and so does the fluorescence. This fluorescence is detected and measured by the PCR instrument, giving us a real-time reading of the DNA amplification process. So, the more DNA there is in the sample, the brighter the fluorescence, and the earlier it will be detected in the PCR cycles. This allows for a precise quantification of the starting amount of DNA, making real-time PCR a powerful tool for many applications in research and diagnostics. As we have seen, real-time PCR is a fascinating process, but how do we interpret the results? Interpreting results from real-time PCR is an art in itself. It's all about the detection and understanding of the fluorescence produced by a reporter molecule. As the PCR reaction progresses, the fluorescence increases, providing us with a real-time insight into the amplification process. But how does that relate to the amount of PCR product? Well, the key is in the correlation. The fluorescence is directly proportional to the amount of PCR product at each stage of the reaction. This means that a higher fluorescence indicates a higher quantity of PCR product. By monitoring this fluorescence, we can quantify the DNA, cDNA, or RNA in our sample with great precision. Furthermore, this technique allows us to observe the reaction at any point, giving us a real-time understanding of the process. This is the beauty of real-time PCR. Interpreting results from real-time PCR can give us precise and accurate data. Real-time PCR is indeed a powerful tool in molecular biology. We've journeyed together through the intricacies of this technique, understanding its underlying principle, the process, and how to interpret the results. In essence, real-time PCR, also known as quantitative PCR, allows us to monitor the amplification of DNA sequences in real time. This is a significant leap from the conventional PCR method, where analysis only takes place after the process is completed. The real-time aspect of this method not only saves time, but also provides a quantifiable measure of the PCR product. The process hinges on a pair of primers complementary to the DNA sequence of interest. As the DNA polymerase extends these primers, the resulting amplicons are re-amplified, leading to an exponential amplification of the DNA molecules. With real-time PCR, we can monitor the progress of a PCR reaction in real time and obtain quantifiable results. It's a technique that has revolutionized molecular biology.